has come. I can see the preview. Alright, we can go live now. Alright, going live. It's good. Okay. Well, I think we are live now. So, welcome back, guys. Today we're looking at biology paper 2 for the year 2016. So, if you are joined, please let me know in the comment section if you're able to get me loud and clear, so that at least you can be able to tell that at least we are able to start. My voice is quite hoarse, the sore throat, not feeling too well currently, so hopefully things will move smoothly today. We had a few challenges here and there, but I think we'll be okay. Alright, so I have 3 in the chat. Still waiting for 6 more to join so that we can get started. Good evening to you too. Alright, good to know, Edward, you're here. Alright, how's the sound? Are you able to get me loud and clear? How's the video? Alright, we're at 4, 5. Okay, nice and clear. Alright, good evening. Alright, good to know that at least you can get me, guys, and I'm clear on your end. That's good. I'm just waiting for the number to reach 10 so that we can get into the paper. We are currently 5 viewers so far from our 30. Okay. Alright, we are 1 minute into the stream. Okay, that's good. Alright, that's good to hear. Loud and clear. That's very good. Okay. Happy to see you guys too. It's been a while. I hope you've been okay. Alright, I'm simply waiting for the... Alright, we're at 7 now. That's good. Okay, waiting for the rest. I'm just waiting for 10 right now so that we can get started. Yeah. I'm kindly on the low side, not feeling too well, down with the cold, flu. So yeah, hopefully we'll get through it. I really wanted to at least to see you guys around so that you know now I'm still around. Otherwise, just been waiting to actually start the stream. But we'll see how tomorrow goes. If I'm not too well, maybe we might not have a stream. But today I just wanted to push myself so at least we can go through this paper and see what. Alright, I think we are here enough. That's good. Alright, so let's get started. I think we have reached our limit. Alright, so let's move into our question paper. Alright, so we are looking at biology paper 2. So this is our first paper 2 paper for our course currently. Alright, so waiting for the screen to simply change. Alright, I think the screen is here. So we are looking at biology paper 2 for the year 2016 for internal candidates. So this was the paper which was written. So let's go through the questions. I hope we'll be able to still go through and see the rest of the questions. Alright, so we move to question number 1. So question number says, figure 1.1 shows an animal cell as seen under an electron microscope. So it's labeled I, F, H and G, as well as the cytoplasm. So we have our figure 1.1. So A states identify the labeled parts F and D, G. So what are the labeled parts F and G? So you can drop your answer in the chat so that I know the letter and the word. So F and G. So what are the labeled parts, F and G? Very good, guys. So for part F, this is what is known as the rough endoplasmic reticulum. So you can see that on the on our structure here, we can see that on the reticulum we do have some dots on it. So this is the rough endoplasmic reticulum, but we have the other one which is smooth. Then for part G, this is the nuclear membrane. So the nuclear membrane, because this is the one which is actually surrounding the what? The nucleus. So that is the rough endoplasmic reticulum, and G is the nuclear membrane. Excellent work, guys. Yes, that is the correct answer. All right, moving on now to question number B. So question B says, explain the functions of the parts labeled H and I. Okay, so we're looking at part C, H and I. So what's the function of part H and I? Yes, guys. What's the function for H and I? So we have H and we have I. Okay. So H. So H is simply the, which structure is this one? That's the mitochondria, and I is simply the cell membrane. So what's the function of the mitochondria as well as the cell membrane? Yes, guys. What will be the function? All right, very good. Yes. Excellent work, guys. Very good. Yes. So for H, this is the mitochondria. So this is where respiration will actually take place. Excellent work, guys. Then the next part is I, which is the cell membrane. The cell membrane will actually be responsible for controlling things which enter and leave the cell, right? So that's why we say that the cell membrane is selectively what? Permeable. So when you receive such questions where they're telling you to mention the part, so you naming the part itself, mitochondria, will score you a mark, and you mentioning the function also score you another what? Mark, right? So don't just go directly into just mentioning the function. So usually these questions will accompany you mentioning the part and the function of the one which will now score you all the forward mark sign. So mitochondria will score a mark, where respiration takes place, that will also give you a mark. Then you mention to say I is the cell membrane, and it's responsible for controlling things that enter and leave the what? The cell. Then question C says, suggest two cell parts. Very good. So cell parts that would be present in figure 1.5 if it was a plant cell. So if this is a plant cell, what structures are we going to see? Which structures can be there in this part? Yes, guys. So which parts are we going to see here? If figure 1.1 was a plant cell, Very good, guys. Yes, very good. So if this is a plant cell, what parts are going to be present? Very good, guys. Yes, so if this is a plant cell, what we are going to see, we're going to see, of course, a cell wall. Most plant cells have a cell wall. Very good. Then the other part we are going to see is that the cell is going to have what? Chloroplast, right? Because plant cells have chloroplast because they are able to carry out photosynthesis, right? So these are the parts which are going to be available. Another part you could have chosen would have been they have a permanent large vacuole. So that's also another part which we normally have when we are going to get. Okay. So that is the parts which we are going to see there. All right, very good. So next we move on to our next question, which is question number 2. Very good. So question number 2 says the figure 2.1 shows the effect of pH on the rate of the enzyme catalyzed reaction K and M. Okay, so we have our J and we have our AY. So we have reaction K and reaction M. So A says what term is given to pH at T, J and L? Yes, guys. Very good, guys. Yes. So what is the term given to the pH at J and L? Looking forward to seeing your responses. Please keep up the energy in the chat. Very good. Excellent work, guys. Very good. Yes. Excellent work, guys. So here, since we are dealing with the pH, it's going to be optimum pHi. So optimum temperature is when we are dealing with the temperature. So if we are talking about pH, here we are going to actually fail to actually use which one? The optimum pH. So that's why the other one will actually work. All right. Very good. So next question we have is question number B, which says explain why the rate of reaction K and M goes down after point J and L respectively. Okay, so why is the reaction going down after the optimum temperature? Yes, guys. Why is the reaction going down after the optimum pH? That's very good. Yes. Please keep the chat alive. Keep the answers coming. Very good. Excellent work, guys. So here what will basically happen is that once we reach our optimum pH, what will happen is that the rate of reaction starts reducing because the enzymes get denatured altogether. So they get denatured after the optimum optimum pH. Again, I'm also talking about temperature, so we usually make a mistake between optimum pH and optimum temperature. So we are saying that here the enzymes start to get denatured because each enzyme has got a specific pHi. So the enzymes here end up getting denatured where the shape changes. Just like, for example, when you get milk and you squeeze a lemon inside the milk, you start to see that the milk starts forming those lumps because the lactogen or the carcinogen starts now coagulating. So that forms those coagulants. So that's why, because the pH changes the shape of the protein. So when the shape changes, we refer to that as being denatured. So each enzyme has got its own specific pHi. So that now brings us to our next question, question C, which says name the regions of the alimentary canal where reactions K and M take place. So where in the alimentary canal are we going to have reaction M and reaction K? So K was in acidic and M was in alkaline. Yes, excellent work, guys. Very good. Excellent. So where we are going to find the acidic environment for K? K is working in acidic environment, so acidic environment will be in the stomach altogether. The M is working in an alkaline condition, so the alkaline condition is actually found in the duodenum. So these are the two regions where we are going to find the reaction K and the M. We have the stomach as well as the duodenum. Excellent work, guys. Yes, those are the correct answers. Well done, guys. All right, moving on to question number D1. So suggest one enzyme which can catalyze the reaction M. So we are talking about enzymes which will be working in the duodenum. Okay, then so here which enzyme will be catalyzing? So the enzymes which work in the duodenum will basically come from the pancreas. So which enzymes do we have in the pancreas? Okay, so which enzymes do we have in the pancreas? Very good. So the enzymes which we have in the pancreas, we have pancreatic amylase, we have trypsin, as well as the lipase. But here I'll simply write our pancreatic amylase. Very good. So that is the one which you have. So pancreatic amylase is one of the enzymes, but you can also go for lipase, you can also go for trypsin, you can also go for what? Which one? Trypsin and the lipase. Then 2, it says state one food nutrient which can be catalyzed by reaction Ki. So reaction K is taking place in the stomach altogether. So since reaction K is taking place in the stomach, so which food is digested in the stomach? Yes, guys. So which food will be digested in the stomach? Yeah, excellent work, guys. Yes, so in the stomach we digest the proteins. So proteins are the ones which are digested in the proteins, because that's where you are going to find the enzyme pepsin. Very good. So that is for reaction K, which is taking place in the stomach. Then we go to part E, which says state 2 factors other than pH which affect enzyme activity. Yeah, so what else can affect enzyme activity apart from pH? Very good. Yes, meat also counts as protein. Yes, guys. Which 2 factors will affect the rate of enzyme activity apart from pH? So we have quite a number now together. So here, very good. Yes, we are right. So what we can use here is that one of the factors is of course temperature. Obviously from the start you guys were talking about optimum temperature. So yes, temperature will be one of the factors. So the higher the temperature, the faster the rate of the reaction before the optimum temperature. So another factor will be enzyme concentration. So when you increase the enzyme concentration, the reaction of enzymes also become faster. Also you can go for substrate concentration, is also another factor altogether. You can also go for enzyme inhibitors, also factors which affect the rate of enzyme reaction. So these were the answers to question number 2, which was worth 9 marks. So if you answered it that way, you would have scored all the marks. So hope you enjoyed this question. Very good. So in case there's any question you want to ask, feel free to leave it in the chat. So we can move on to the next question, which is question number 3. Okay, so we have question 3 now. So question 3 reads, figure 1.1 shows a cross section through a stem of a plant which had been previously dipped in a red dye for 6 hours. So we have a red dye and we have dipped this for 6 hours. So the diagram is labeled the N, O, P, Q, and R. So question A1 says identify the parts labeled the N, P, and the Q. So yes, guys, which do you think are the parts now for part P, Q, and the R? So N, P, and Q. Very good, guys. Yes. So what is N, and what is part P, and what is part Q? Very good. Yes. Excellent work, guys. Very good. So for part N, excellent, guys, this is the cambium. Very good. So part N is the cambium. How about part P and Q? Very good. Excellent work. So Q will be the phloem vessel. Very good. And the So P will be the phloem vessel and Q will be the xylem what vessels altogether. So these are the parts there. So O is the cortex. So the cortex is actually OI. Very good. So that's O will be the pith. Then for R that will be the cortex. So that is the parts of the stem. Right? So we have the pith, cambium, phloem, xylem, as well as the cortex. So those are the correct answers. Very good, guys. Well done. Excellent. So let's move on to B now. I hope you'll still be able to remember our diagram. Okay. So 2 says which labeled part in figure 1.5 will be stained red. So which part is going to be stained red? Altogether. So which labeled part is going to be stained red after 6 hours? Yes, guys, which part will be stained red? Excellent work, guys. So the part which will be stained red will be Q, because the Q, that is the xylem vessel. Right? So B says, 3 says give a reason for your answer in A1 above. Okay, so what will be the reason? Why is it going to be stained red? Very good. So it's going to be stained red. Excellent way, guys, because the xylem vessels are the ones which transport water and mineral salts, meaning that the dye itself will be acting as a mineral salt. So that will be the one which will be carried inside the xylem vessels. So the xylem vessels transport water and mineral salts, so they're the ones which are going to be stained. Excellent work, guys. All right, so next question we have is question number B. And question B says suggest 3 factors that would increase the rate of movement of the dye in the stem when it's dipped in red dye. Okay, so we're looking for 3 factors. So what factors cause the water to move up the plant faster? Okay, yes, guys, so we've been looking at this question a lot of times, and I've been talking about this one a lot. Yes, guys. So which factors will affect will increase the movement of water of the dye in the plant? So this question is because remember that for you to absorb water you need transpiration, isn't it? So this question is again asking us to say which factors affect the rate of transpiration, or which factors make transpiration take place faster. So I know you might be wondering about, so now, very good, yes, we are talking about humidity, transport, what now we want the temperature. So which factors are we going to work? So don't just say temperature, humidity, you have to mention to say you need the high temperature altogether. So you are going to need high what? Temperature. The part of high temperature, what are you going to need? High light intensity. So apart from high light intensity, you also need low humidity. I always emphasize on this one to say humidity is the only factor which you require to be low for you to have a high rate of transpiration or high absorption of water. Here you could also have gone for wind speed, more wind speed. So you could have just gone for wind. So all these are the factors which affect transpiration. Well done, guys. That was very good. All right, so moving on to question number 2, it says name the processes that will cause the movement of red dye in the stem. So name the process that will cause the movement of red dye in the stem. So now we want the process. Okay, so which process will now cause the movement? What do we call the process which causes water to move up the plant? Excellent way, guys. Yes, very good. This is transpiration pool. Excellent. So transpiration pool is what will cause the water to actually move up the plant. Excellent work, guys. So these were the answers to question number 3, and this was worth 9 marks. So before I move on, just a tip to remember on question number, especially for 1 and 2. So these two questions are tied. Now the problem when usually find that if you get this part wrong, even the next following one will also be wrong. So there's no way you can get Q, you mention N, and then here you write xylem. So both will actually be marked wrong. So you have to be careful in answering such questions which give a follow-up to say from the answer above. So you have to be careful on the one you give above, so that the one which follows is also tied to the other one as well. Okay, so we'll move on now to question number 4. All right, so we are on question 4 now. So question 4 says figure 1.1 shows a certain type of flower. So we have a flower there with the paleo, which is the bracket. Then we have parts labeled S, T, and U, and this is our figure 4.19. So A says identify the parts labeled S, T, and U. So what are the parts S, T, and UI? Very good, guys. Yes, keep the answers coming. Yes, guys. Yes, guys. What are the parts S, T, and U? Yes, guys. So we have figure 4.1, which is showing a certain type of flower. Oh, sorry to hear about the network. I know with the cold weather, especially the way network is, hopefully the signal will pick up. Let me just see. But from here, the transmission is telling me it's not that bad. Okay, I have green in terms of transmission. Okay, maybe to pick up on your end that side. Okay. All right, very good. Okay. Excellent work, guys. So yes, so S is the anther. So that is where pollen grains are formed, and the T is the feathery stigma, very good, of the flower, and the U will be the ovary where the ovules are contained. So that would score you 3 marks. Excellent work, guys. So I can see we know the parts of the flower, which is excellent work. So we have the S, which is the anther, T, which is the stigma, and U, which is the ovary. All right. So next we move on to question number B. So question B says here, B1 says identify the type of pollination that occurs in the flower in figure 1.1. So which type of pollination are we talking about here? What would be the type of pollination of our nice flower here? Yes, guys. Which type of pollination? Excellent work, guys. Very good. So the type of pollination which we have here is wind pollination. Excellent work, guys. So we have wind pollination. So here the question is asking us to say, give 2 features from figure 1.1 which supports your answer in B1 above. So which features have we seen? Which are the adaptations of a wind pollinated flower? So just 2 adaptations of a wind pollinated flower. So we looked at this one the time we were looking at the topic reproduction in sexual reproduction in flowering plants. I hope you remember that lesson. So what are the two adaptations? So here they're just asking us for two. Yes, guys, very good. So one of the adaptations which we have is that we have long pendulous filaments. So the pendulous filaments are long. You can see they are hanging outside the flower. So then the other adaptations which we can notice there is that you have a feathery stigma. So we have a feathery stigma which is present. So apart from that, we can also mention to say the anthers are larger because they produce more pollen grain to increase the chances of pollination. Then, of course, we could have said that it has a bracket, mean that it has no colored petals. So that is the other adaptation which we can use here. So these were the answers to part B. Moving on to part C now. So part C says, 1. State one other type of pollination other than the one mentioned in B1 above. So we mentioned the wind pollination. So which is the other type of pollination which is available apart from wind pollination? Yes, guys. Which is the other type of pollination? Very good, guys. So the other type of pollination which we have is the insect pollination, where the flowers are pollinated by what? Insects. Very good. So we move on now to question number C2. C2 says, give two characteristics of the flower where the pollination mentioned in C1 above occurs. So we're looking at characteristics, if we're looking at adaptations of the flower to insect pollination. So what adaptations do we have in insect pollinated? So previously we were answering on the adaptations of a wind pollinated flower. So what adaptations do we have now for an insect pollinated flower? Yes, guys. Yes, guys. Very good. So for insect pollinated flowers, we can simply say they have brightly colored petals to actually attract what? Insects. So we are going to see that these flowers are going to be brightly what? Colored. Very true. Then the other adaptations is that they will actually have nectaries to reward what? Insects. So here you can say they are brightly colored, they are scented. All that is to attract what? Insects. Then apart from that, they will have nectaries to reward what? Insects. So that is what the question was asking us for. So I know the question was asking for types and agents, but the question in this case was actually talking about another aspect of pollination. So that's why we find that in most cases for this question, we find that the answers which were related to was for insect pollination and the wind pollination. So the examiner wanted to find out whether the candidates are able to distinguish the adaptations of the insect pollinated flowers as well as the wind pollinated flowers, and also to mention the adaptations. So that was what was required of this question. Very good. Yes, so the bright colors and scent, that is to attract insects. So that's why you have to be careful when you're wearing perfume. When you wear perfume which is smelling like roses, don't pass near beehives, otherwise the bees will actually follow you and think you are actually a flower. So don't be surprised when bees start following you after you have sprayed your perfume, because they actually think you are a flower and they are looking for nectar to actually enjoy from the flowers. So nectaries are the part of the flower which contains the sugar. I don't know if you guys, you are too much of being in my yard, but for us guys, When we are growing up, to get some flowers, you do pull it out and start sucking on the flowers. So I don't know if any of you in the comments have ever done that before, where when you suck on the flower, you actually feel that the flower actually tastes what? Sweet. So those are the nectaries, they contain sugar. All right, so that is question number 3. All right, so we move on now to question number 5. So we have question 5 now, which of course is always genetics in biology. So always expect that you're going to have genetics for question 5, unless maybe ECZ just decides to change, but it's always been tradition that question number 5 has always been under genetics. So figure 5.1 below shows a pedigree diagram for 2 families A and B, outline the inheritance of a sex-linked disease called the hemophilia. So we have family A, family B, and we have parents, and we have offsprings H and J. So the key is that the unshaded circle is normal female, the unshaded square is male, the shaded circle is hemophilic female, the shaded square is hemophilic male. So A1 says, from figure 5.1, which family has a parent who is a carrier for hemophilia? So between family A and B, which family has a carrier for hemophilia? Is it family A or family B? All right. Yes, guys. Very good, guys. Excellent here. So the one which will be a carrier for hemophilia is family A. Very good. So in family A, we have a carrier for what? Hemophilia. Excellent work, guys. So that is where we are going to find a carrier. All right. So carriers appear normal, but they have a genetic disorder. So those are the ones referred to as carriers. Very good. All right. So that brings us back to question number 2. Okay, so question number 2, 2A says, give a reason for your answer in A1 above. Oh, due to my over talking, I think I've even given the answer already. All right, so family A, we are saying has a carrier. So what reason have we given? So what's the reason for us choosing to say we have chosen family A as the carrier? All right, very good, guys. So family A has a child that is hemophilic, so hence the gene was passed by a carrier. So we are saying that we have chosen family A, the family has a child that is hemophilic, so hence there is a gene which was passed by a carrier. So for you to have a disorder, it means that someone had passed on the gene. All right, so we go to B1 now. So using the symbols XH and the X small letter, state the genotypes of the offspring HJ if J is a carrier for hemophilia. So J will also be a carrier. So they're saying J will be a carrier for what? Hemophilia. Yes, guys, what is going to be the answer here? I know these are going to be difficult for you guys to type, so we'll simply move on fast so that we don't actually have a challenge. Okay, this one. Okay, so this will be hemophilic. Yes, guys. So I just want you to tell me how the genotype is going to be. Yes, guys. Okay, so we're almost there. Alright, I'm almost done. Okay, so I'll just say entering the numbers a bit. Okay. Alright, so we are done. Okay. So we have said that the child has hemophiliac, so hence the genes will be passed on by a carrier. So H has got hemophilia, so the genotype here, that would be a hemophilic male altogether. Then we are told that J is a carrier altogether. So a carrier meaning that they are going to have both a normal gene and a hemophilic what gene. So they have a normal gene, so this gene is for normal, and this gene is for hemophilia. So H has got hemophilia, so that they've got the hemophilic gene, which they got from their carrier mother altogether. So these are the genotypes. So next, now that we have the genotypes, we can now move on to using a genetic diagram to show whether the offspring will be hemophilic or normal when H and J are crossed. So we are crossing H, hemophilic male, and the carrier female. So let's see what the cross is going to be. So whenever you're going to carry out a genetic cross, first thing you need to do is to write the mother and the father. So these are the ones who are going to do the what? The crossing. So once you've written the mother and father, there are 5 steps which you have to write under a genetic what? Cross. Okay? All together. Very good. So once you are done with that one, first thing we write now is the parental phenotype. So we have said that the father is hemophilic and the mother is a normal. The mother is normal, but the mother is a carrier. So that will be step number 1. So make sure you write this word nicely. Everything should be there. Parental phenotype. Then the genotype of the father, hemophilic crossing with the normal. So this X has to be there. If you leave out the X, then step number 1 will be marked wrong. So the X has to be there to show that mating is taking what? Place. Then next, once you are done with this step, you go to step number 2, where you now write the parental genotype. So you now put in the genotypes. So we've said the H, this was the genotype which we got, where hemophilic, they've got a hemophilic gene. Then the mother is normal, so J is a carrier, meaning that they've got a carrier gene. So once you are done with the parental genotype, next step is to write the gametes. So when you come to the gametes, the only thing you simply do is to spread the what? The genes apart. Then if you want, you can put them in circles. So if you want, you can say, okay, this one in a circle, this one in a circle, but it's optional if you want. Okay, then also this one in a circle to show that they are gametes. The next, you now start crossing to say this one will cross with this one altogether. Then this one will cross with this one. Okay, then this one will cross with that one. Then this one will cross with that one. So that will now move us to the next step, which is to write the F2 offspring genotype. So the hemophilic. So here we're going to have this small x, it has come here. The capital X has come, the capital H has come here. Then here, small h as well. Then here Y and the X. So that gives us our phenotype. So next step is to write the F2 offspring phenotype. How are they going to appear? So here, our first one is going to be normal. So this will be a normal female. Okay, so we have normal female because there's a normal gene in here. So we have a normal gene, which will overshadow the hemophilic. So this female is going to be normal, but they will be referred to as a carrier. This female has got 2 hemophilic genes, so this female will be a hemophilic female. Then the next one we have a male who has got a capital H, so this one, since they've got a normal gene, this will be a normal male. Then the next one will be a male who has got a small the hemophilic gene, so this will be a hemophilic what male. So from the cross we can see that the children will be, you are going to have normal as well as the hemophilic. So normal you have 2, and the hemophilic you also have 2 chances. So here the ratio is 1 to 1, so there's a 50% chance of getting a normal child and a 50% chance of getting a hemophilic child. So this is how you show a genetic cross. Now most people fail to write a genetic cross because most of you just write it in shortcuts. These words which I've written down here, you normally do not write the parental phenotype, parental genotype, gametes, F1 or F2, depending on which generation your cross is. So this is we are the parents first, then we are the offspring, then they are crosses. So this is the F2 generation. So F2 offspring generation you also write, F2 offspring genotype you also write. So these steps are very important for you to write them. Otherwise, if you just see cross without writing these stages, everything will be marked wrong. So this is 5 marks, mean that you get a mark for parental phenotype, a mark for the parental genotype, a mark for the gametes, you get a mark for the offspring genotype, and a mark for the offspring phenotype. So that is where the 5 marks comes from. So without you writing these words on this side of the cross, will actually lead you not to score on the genetic cross. So even if you cross nicely without writing the titles of the cross, you won't be able to score anything. So that is why genetics is the most simplest question, and it's also the most difficult question to actually score altogether. So that is how we normally get that one. So I didn't write F2 because remember we have the first parents, which is family A, so that is the first level of parent type. So then when they have children which are H, so H are the F1 altogether. Then when F1 marry and have children, they now become F what? F2I. So that is how they move. So if F2 again marry and have children, they'll be known as F3. So that is how it moves in. Okay. Alright, so that is our question 5. Alright, so we are done with section A. Now we move on to section B. So this is where we have essay questions now together. So we are now moving to essay questions now. So essay questions have 36 marks. So here you're supposed to answer any 3 questions in this section. So any 3. So here all answers must be in complete sentences and the paragraphs. So here there are no bulletings, so don't put any bullet points. So all your answers should be in complete sentences and the paragraph. So we start with question number 6A, which says explain gaseous exchange in green plants. 1 during the day, 2 at night. 4 marks. Then B says using named organisms explain the industrial application of respiration. 4 marks. Then C, describe how gum disease is brought about. Also 4 marks. Now together. So mean that here we have to present our answer in complete sentences and the paragraphs. Alright, so we go to question number 6A, which says explain the gaseous exchange in green plants during the day and at night. So during the day, how is our gaseous exchange, and at night, how is gaseous exchange? So what are we taking in during the day, and what are we taking out at night? So during the day, what do we take in and what do we take out? And at night, what is taken in and what is taken out? Yes guys, so how is gaseous exchange? during the day and how is it at night? All right, very good. Okay, so during the day we are going to have two processes taking place. We are going to have photosynthesis and we are going to have respiration as well taking what place, right? So during photosynthesis, the plants will take in they'll take in carbon dioxide, which they will react with water to produce glucose and the oxygen. Right? Then the oxygen which is produced, some of it will be used for respiration to produce energy and the carbon dioxide. So the carbon dioxide which is produced is again used for photosynthesis. Then any extra oxygen which is produced will then be coming out of the leaves as the carbon dioxide. Then when you come to night time, at night only respiration will take what place. So at night only respiration takes place, so mean that plants will take in oxygen and give out carbon dioxide. So this goes in, this goes out. So that's why it's advisable that at night do not sleep with plants at night, because they'll also be using up the oxygen which you have in the room. So how do we present this answer now? So to present this answer, what we simply say is, number one, during the day, as photosynthesis takes place, which is uses carbon dioxide and produces the oxygen, this makes carbon dioxide to be at low concentration inside the cell and at high concentration in the air spaces surrounding the cells. So carbon dioxide here, what will happen? It will diffuse into the leaf through the stomata, where it is at low concentration. It then diffuses into the moist, spongy mesophyll and into the adjacent cells. Then oxygen produced during photosynthesis is used for respiration. Then the excess oxygen diffuses out into the atmosphere through the stomata. So that is how gaseous exchange will be taking place during the day. So the main important thing to notice is that during the day we have photosynthesis which is taking place. Then now when you come to the night time, so at night time no photosynthesis takes place, only respiration will take place, which uses oxygen and produces carbon dioxide. So carbon dioxide is in high concentration, so this causes oxygen to diffuse into the leaf mesophyll, where carbon dioxide diffuses out of the leaves through the stomata again. So this is what happens during the night and what happens during the day. So during the day we only have photosynthesis and the respiration taking place. Okay, so but at night no photosynthesis takes place, only respiration. So this is the solution to question number 6C, A1 and 2. Moving on to 6B. 6B says, using named organisms, explain industrial applications of respiration. So which industries use respiration? Yes, guys, very good, very good. So that was a good explanation on that part. So which industries will use respiration? So no need for you to write here, just want the points which we are going to discuss on this part. So we're talking about the industrial use of respiration and which organism are we using for that process. Okay, very good. So for respiration, the industrial application we are going to use yeast for brewing. So yeast is used for brewing because when you have anaerobic respiration, it produces carbon dioxide and alcohol, which produces the alcohol for alcoholic beverages. Then the another application is that we also use yeast for baking altogether. So when baking, yeast is added to the dough, which carries out the anaerobic respiration, producing alcohol and carbon dioxide. So that carbon dioxide is used for raising the dough. That's why I find that people are making bread or making fritters, when they leave the flour for some time after adding the yeast, you find that the flour will start rising. we call that the duff. I know we normally refer to it as flour, but once you mix flour, water, sugar, we call it the duff. But normally we always be saying flour, but okay, something to remember about that point. Okay, so the other application is that we also need the lactobacillus, which is bacteria. So you need lactobacillus for production of cheese and yogurt altogether, because this is the one which will carry out anaerobic respiration, producing lactic acid. So it's the lactic acid which gives the sour milk its sour taste, that helps coagulate the proteins, and that is the protein which is collected and hardened to now form the cheese. Okay, so then another application is that we also use anaerobic respiration bacteria in sewage treatment. So this is where we use the industrial applications of respiration. So we need it in brewing, in baking, in dairy products, as well as this sewage treatment. This is where respiration is used industrially. Okay, so how do we present our answer now? So we are saying respiration is used in the following industries: baking, brewing, dairy, and sewage treatment. Then we explain, say, in baking, yeast produces carbon dioxide, which causes the dough to rise. In brewing, yeast produces alcohol during anaerobic respiration. Then in dairy, anaerobic bacteria produces lactic acid altogether, which gives milk its sour taste and coagulates carcinogen. Then in sewage treatment, anaerobic bacteria turns the organic waste into harmless substance that can be used as organic fertilizers altogether. So that is used as night manure, because people used to get this manure at night to go and die, so that's why it's been given the name night manure. And also releases methane gas. So methane gas can be used as a fuel for lighting gas stoves and also producing electricity. So that's why if you have a town, make sure you make use of the sewage from the town, because that same sewage, you will actually add more money back to the town itself. So don't let just the sewage be thrown away, because the sewage can actually be used as fertilizer to grow more crops, which people again need to eat, and also you need it for fuel, because people don't use charcoal. So from the same waste material, methane gas can be produced. So this is how you present your answer for question number B. So you can see here I've not used any bullet points. Everything has been given in a nice paragraph, right? So that is how the answer should be written. So moving on to the next part of the question, which is question number C, which says describe how gum disease is brought about. So how is gum disease caused? Yes, guys, very good. So gum disease, very good, will be caused by bacteria altogether. So when the bacteria actually in the mouth will form plaque, and once that plaque hardens, the bacteria will then infect the gums. So that is what is known as gum disease. So you find that in this case, the person's teeth, instead of them being covered by the gum on top, so instead of all this part of the teeth being covered by the gum, you can see that majority of the teeth is actually being exposed. So this is what is referred to as gum disease. So all this part was supposed to be covered by the gum, but because of the plaque, the gum is actually becoming slower and slower. So that will actually cause the teeth to start falling out, and that leads to swelling of the gums. So we can simply say that here for C, gum disease is a very common dental disease. It is caused by bacterial infection. So we have bacterial infection. So when food accumulates, it builds up and forms a hard layer called plaque. So plaque can be removed by brushing, but if left to harden, it cannot be brushed or fine. So bacteria in plaque will then infect the gums. So once the plaque bacteria infects the gums and it causes them to swell up and bleeding when brushing, the condition is called the periodontal disease, or simply gum disease. So this is how gum disease is caused. So you give your answer in that way, you are able to score all the nice 12 marks. But if you give your answers in bullet points, where you are saying bullet, so everything, even if your points are correct, everything will be marked wrong. So this is something you should be careful on when you come to section B. All right, so moving on to question number 7. Okay, so we have question 7 now. 7a says describes the function and the structure and functions of endocrine glands. Then 2, explain the differences between the endocrine gland and the salivary gland. Then B, explain the function of the following hormones in the body. A, insulin. B, antidiuretic hormone. Both 3 marks. Okay, so we'll start with question number 7a1, which says describe the function and structure of endocrine glands. So how is the structure and function of endocrine glands? Okay, all right, I've taken note. Okay, I'll use the red pen then. Okay, very good. So when you come to endocrine glands, so these are the endocrine glands which we have in our body altogether, and these glands are very important because they are what referred to as the ductless glands. So these glands will form what is known as the endocrine system. So these glands produce hormones, which is a chemical which is transmitted in blood altogether, and they have no ducts, so that's why they are referred to as ductless glands. So the hormone will be carried in blood and it's destroyed by the liver, and they will affect different parts of the body which contain receptors for that hormone. So glands which we have in the body, we have the pituitary gland, so we have the pituitary gland, we have the thyroid gland, this is the thymus. Okay, we looked at this. Then we have the adrenal. Okay, then we have the pancreas. Okay, then we have the ovary. Okay, then we have the testes. So these are the glands which we have. So let's describe now the functions which we have. Okay, so A1 says endocrine glands will form the adrenal system. So this is the system of coordination which is made up of ductless glands. So that produce what? Hormones. So a hormone is a chemical secreted by ductless glands, transported by blood, and affects one or more target organs before being destroyed by the what? The liver. So a target organ is any organ that carries an appropriate response to a stimuli under the influence of the hormone. So any hormone can only have effect on an organ that has receptor sites for it. So that is how we describe our adrenal glands. So we are saying adrenal glands are ductless, they produce hormones together, and these hormones will be transported in what? In blood, then affect one or more target organs before being destroyed by the what? The liver. Then a target organ is any organ that carries out an appropriate response to a stimulus under the influence of hormones. So any hormone can have effect on any organ that has receptor sites. So if there are no receptors, then the hormone will have no what? Effect. So that answers our question number A19. Very good, guys. All right, moving on to A2. A2 says explain the difference between adrenal glands and the salivary glands. So what's the difference, guys, between the adrenal gland and the salivary gland? Okay, so for this one, the question does not require you to draw altogether. So I'm just giving you this drawing so that we don't feel bored. Otherwise, I would have just gone directly to just writing the essays. But I just wanted to present these guys so that you can see them visually, so that when the answers come, you're able to understand. Right? So otherwise, if the question doesn't tell you to draw, no need for you to draw. Right? Okay. All right. So moving on. So differences between the salivary glands and the adrenal glands. So you can see here we have the salivary glands. So the salivary glands are found where? In the mouth. Now together. While these ones are found where? On the kidney. That's where we find the adrenal glands. So these ones have got tubes. You can see here there's a tube. So mean that these ones have got ducts. But these have no tubes. So these ones are ductless. So these ones will produce saliva. Okay, while these ones will produce hormones. So they produce hormones. There are 3 hormones which it produces. We have adrenaline. okay, adrenaline, we have cortisol, okay, and we also have aldosterone, which is used for balancing sugar levels. So these are the differences, eh? So let's now present our differences in an essay. So we are saying adrenal glands and salivary glands have the following differences, okay? Adrenal glands are located on the kidneys altogether. So why was I writing will? It was supposed to be while. All right, let me edit this. Yeah, what was happening? So it's supposed to be, so when we're differentiating, we use the word while. So let me just edit my writing here. Oh, I didn't see that one. Okay, where am I? Okay, kidneys. Okay, so here it's supposed to be while. Okay, what else? Okay, delete this. While salivary glands are located in the mouth, then adrenal glands are ductless, while salivary glands have salivary ducts. Okay, then we say salivary glands secrete saliva, while the adrenal glands secrete adrenaline, cortisol, and the aldosterone hormones. Okay, so that is how we differentiate. Okay, so now when you come to differentiation, okay, so we are saying adrenal glands and the salivary glands have the following differences. So we have adrenal glands are located on the kidneys, while salivary glands are located in the mouth. Then adrenal glands are ductless, while salivary glands have got ducts. The salivary glands secrete saliva, while adrenal glands secrete adrenaline, cortisol, and the aldosterone hormones. Now, when you come to differentiating, always make sure that you are differentiating one point at a time. So if you talk about location, just talk about location, then full stop altogether. Then you come to another point, also differentiate that point on its own altogether. So that's how you're able to score the marks. So don't group them together to say adrenalines are located on the kidneys, they are ductless, and they produce hormones. Then you say while once. No, you have to write each sentence, each point on its on its own. I hope I'm being clear on that point, so that you're able to score each sentence on its on its own. Otherwise, when you group it, it's difficult for the examiners to actually give you those 3 marks which are needed, right? So that's why it's very important that whenever you differentiate, pick a point and finish it, then full stop. Another one, pick it. I know it feels like repetition, but you just have to repeat. That is how you'll be able to secure all your all your marks, right? All right, so that puts an end to our question number A. We now move on to question number B. So B says, explain the function of the following hormones: insulin and the antidiuretic hormone. So what's the function of insulin and what's the function of antidiuretic hormone, or simply ADH? All right, very good, guys. Yes, so insulin, this one is a hormone which is secreted by the beta cells of the pancreas to actually produce to lower the glucose levels. Then the antidiuretic hormone is secreted by the pituitary gland to increase the reabsorption of water in the kidneys. All right, so let's see how we are going to write this essay. So we say B1, the beta cells of the pancreas secrete the hormone insulin when glucose levels are too high in blood. So the pancreas detects the changes and secretes insulin, which lowers the glucose levels in the following ways. So how does it do it? It causes the cells of the liver and muscles to convert excess glucose into glycogen altogether, which is stored in the liver and muscle cells. So the body can only store about 400 grams of glycogen. So we are talking about say 100 grams in the liver and 300 grams in the muscles. So it will also cause the cells of the adipose tissue to convert glucose to what? To fats. So that's why it's very important to notice this point, is that for the fat which you have in your body, actually majority of the fat does not come from eating meat altogether or eating cooking oil. So it's actually the sugar which we are eating which actually makes us what? Fat altogether. So that's why you're going to find that it's actually the enzyme which will make you fat and not the relish altogether. Because the more lumps of enzyme you eat, if you're not using the glucose, majority of that enzyme will be turned into what? Into fats altogether. So normally people will be blaming the cooking oil to say, no, if vein is a salad, actually it's the enzyme which actually makes you fat. So the enzyme, the potatoes altogether, the ice cream, the chocolate is what actually leads to you gaining what? Weight. That is something to just keep in mind. Okay, so if you want to lose weight, reduce on the lumps of enzyme you are eating and increase the number of vegetables. Okay. So the fats are stored under the skin and under delicate organs such as the heart. So the more fats you have around your heart, it ends up leading to heart attack. But in normal cases, the heart, liver, kidneys, intestines, and brains need the fat to act as a shock absorber. So it will enable body cells to also absorb glucose from the blood and they use it for respiration. So this is how insulin helps the body reduce glucose levels. One, we're talking about changing glucose to glycogen, and any excess glucose which has not been turned into glycogen will be converted to fats altogether. So then it also makes the cells take up sugar and they use it. Then when you come to the number 2, which is the antidiuretic hormone. So antidiuretic hormone is also known as vasopressin. So the hormone is stimulates the reabsorption of it stimulates the reabsorption of water from the renal tube and the loop of Henle in the kidneys. So when the body has little water. So if you don't produce enough ADH, it leads to a condition known as diabetes insipidus, which is a condition where a person will actually pass out large volumes of dilute urine. So a person will constantly be urinating altogether. So they'll keep on urinating, but most of the water will just be urinated out of their bodies. So this is the role which these two hormones work. So insulin will lower the glucose levels. Then the antidiuretic hormone is used for reabsorption of water in the kidneys when there's little water in your body. So bear in mind how these two hormones will work. All right. So next we move on to question number 10. Oh, sorry, no, why 10? Sorry, I still want to finish with this. Is question number 8. So question 8 says, explain how HIV can be transmitted from one person to another. Then B, 1, explain the causes of stigma to a person living with HIV and AIDS. Then 2, describe ways of reducing stigma. All right, so we go to question number A. How is HIV and AIDS transmitted? Yes, guys. So how do we transmit HIV and AIDS? Oh, flu is catching up on me now. Yes, very good, guys. Excellent work. Well done, guys. That's very good. Yes. So when you come to transmission of HIV and AIDS, make sure you include that this is occurring from an infected person. So the first way is, of course, unprotected sexual intercourse with an infected person. Then we can also get HIV from sharing sharp objects like endodermal needles with an infected person. Altogether, HIV can also be transmitted through blood donation from an infected donor. So if the donor is infected, you find that in most cases that's why blood donation normally you might not need to it needs to be scanned. Then the other way in which it can be transmitted is, of course, through a pregnant mother who's an infected mother to child during childbirth altogether. and it can also be transmitted through breastfeeding mother who's infected to infant if they are breastfeeding after 6 months. So this is how HIV and AIDS can be transmitted. So in all the options, make sure you indicate that either the mother is infected, the partner is also infected. Otherwise, if you don't mention that any of the individuals infected, no transmission will take place. Then be careful also when you come to sexual intercourse. So I mentioned that its transmission will occur through unprotected sexual intercourse with an infected person. So here do not write that no by sleeping with a person who has HIV. No, because in biology sleeping is sleeping. So you can't get AIDS from sleeping, because when you're sleeping, you're just sleeping. You can't get HIV and AIDS. So you mentioned that it's unprotected sexual intercourse. Apart from that, again, don't just write having sex, because sex is the state of gender, male or female, so it has to be sexual intercourse. Then next we say sharing sharp instruments such as endodermal needles, laser blades, toothbrush with an infected person. Apart from that, contaminated blood transfusion from an infected person. Then also transmission from an infected mother to fetus during pregnancy or delivery, and the transmission through breastfeeding after 6 months by an infected lactating mother, and also organ transplant from an infected donor. So that's why we have to be cautious here when you're basically talking about the transmission itself. Okay, very good. So unfortunately here when we come to the issue of kissing, so kissing will be a bit difficult to actually, because there are different types of kissing. So here the points which we use are normally these ones. So those might fall under myth side. Okay. All right. Thank you so much for that remedy. I'll try it as well. Okay. Thank you guys. Okay. Hopefully I'll be better soon. Okay. All right, next we move on to question number B1, which says explain the causes of people living with this stigma with HIV. Okay, so what will cause it? So what causes stigma to people living with HIV and AIDS? So why are people afraid of people who have HIV and HIV? Yes, guys? So why is it that we are afraid of people who have HIV? Why are people afraid? Why do they stigmatize? Yes, very good. Very good. Yes. So one of the reasons why people end up stigmatizing people living with HIV and AIDS is simply because of myths. So myths, and as well as fear. People are afraid of getting HIV and AIDS. So stigma to people living with HIV is actually caused by the following. One, fear of contracting HIV and AIDS. People are afraid to say, oh, if I get AIDS, I'll be dead altogether. So people have fear. So once they hear, no, that one has AIDS, they will try to run away the opposite direction. Apart from that, also myths of how HIV is transmitted. So for example, what are the myths? People say you can get it through shaking hands or hugging or sitting or even just talking to a person with HIV and AIDS. So this one is not transmitted like where corona is, where it's, as you are talking, particulates are moving. So that is airborne, but HIV is actually sexually transmitted. So that one cannot be obtained by talking to a person with HIV and AIDS. So yes, shaking hands, especially corona, all these, that's why we are saying no handshaking. That's why we are using fist pumps when we are actually touching, because these are pathogenic diseases which can actually be obtained. Yeah, the other cause, of course, is lack of information. So if people don't have information, they'll start now creating false information. That's where my myths come from. So lack of information on the available services for people living with HIV and AIDS. So these are the areas which cause this stigma, fear, myths, and the lack of information. So when people don't have information, they'll start creating their own. So that's why I'm saying rumors are bad. So those are the causes of stigma. All right, so let's now move on and look at the ways of reducing stigma. So how can we reduce stigma to people living with HIV and AIDS. Yes, guys, so ways of reducing stigma. How can we reduce stigma to people living with HIV and AIDS? So ways in which we can reduce stigma is by one, providing a balanced diet to people who have HIV and AIDS. So by ensuring that people with HIV and AIDS have a good balanced diet, so don't say mukufa manje, so no need for you to be eating chicken and chips altogether. So don't be like that. So we have to make sure that people who have HIV and AIDS are always given a good balanced diet. Then apart from that, we also need to make sure they are access to antiretroviral drugs or therapy, so that they are able to actually improve their immune system, so that they're able to live longer. Okay. Then another factor is, of course, through care and support, so that people with HIV and AIDS can actually be able to receive their medical equipment, and also they're able to receive counseling and guidance on how they're able to live longer. And apart from that, we also need to carry out advocacy to inform the public on the dangers of stigmatization, and also the members of the public are aware of how HIV is transmitted. Remember I said that if there's no lack of information, it will lead to stigmatism. So I hope we've been seeing adverts on TV talking about HIV and AIDS. Are they still showing information on HIV and AIDS? Was last time I heard, was it on Facebook, where they were saying, is it 4,000 youths are getting HIV each day? That was quite scary. So that's why we need that information on ZNBC. I know ZNBC is mostly Tredkin's adverts, but at least hopefully they will be sharing more information on HIV and AIDS. So the answer will be presented in this way. Number two, stigma towards people living with HIV and AIDS can be reduced by the following ways. One, giving them a well-balanced diet. Number two, medical treatment by providing ARVs, which slow down the symptoms and illness. Then provide care and support to people with HIV and AIDS, and the advocacy and counseling on HIV and AIDS as well. So these were the solutions to this part of the question. Are we clear on that, guys? Very good. Alright, otherwise, really good to see your answers in the options. That is very good. Please, let's keep it up. Well done, guys. Okay. Alright. So next, now... Okay. Very good. So next we'll go to question number 9. So question number 9 says, A, state the parts of the synovial joint and explain their functions. Number 2, explain why a bone is considered a living tissue. Number C, explain the actions of antagonistic muscles of the eye when one moves from a dark room to a bright room. Okay, so we'll start with question number 9A. State the parts of the synovial joint. So let me know in the comment section, which parts of the synovial joint do you guys know? Yes, guys, which parts of the synovial joint? Oh, my video is gone. Oh, seems you guys, you like seeing me as well. Good to know. Thank you for letting me know the video was gone. Okay. Alright, so which parts of the synovial joint do we know? Alright, very good, guys. So when you come to the synovial joint, there are quite a number of parts. So we have the cartilage, we have parts, of course, we have the bone, we have the cartilage, we have the synovial fluid, we have the synovial membrane, we also have the joint capsule. Then the part which is also missing here is the ligaments altogether. So we also have ligaments which are supposed to hold the bone to the bone. So let's look at how the functions of these parts work. So The cartilage will be preventing friction. So cartilage will prevent friction. Okay, so this one will prevent friction. Okay, then the capsule will hold the joint. Synovial fluid, this one will provide the lubrication so that the bones are moving slowly. The membrane produces the fluid, so this is the one which produces the fluid. Alright, so let's write the essay now. So here we can simply say the synovial joint has the following parts. We have the cartilage. So here we have cartilage. We have the ligaments, synovial fluid, synovial membrane, tendons, and the joint capsule. So by you, excuse me, by you first mentioning the parts and putting a full stop, you're able to score marks on each of the parts which you have written, even before you now write the function. So that's why it's important that you mention the parts, full stop. Then the next, now you start mentioning the functions of each part separate. So next you go now to say cartilage will prevent friction and the grinding of the bones against the bones. And it also acts as a shock absorber. So that is also another point. Then next point, we go to the ligament. Connects bone to bone and prevents dislocation of the joint. Then here we say they also keep the joint stable. So that is also another function. Then the synovial fluid, its function is it's a lubricating fluid that reduces friction in the joint, and it also acts as a shock absorber. So this one will also work as a shock absorber. Then the synovial membrane secretes synovial fluid. Then the tendon's function is to join the muscle to the bone. Okay, so we have tendons which will join the muscle to bone. Ligaments join the bone to bone. So remember about the difference between a tendon and a ligament. So normally I always remember this difference, especially when you are eating meat. Especially I ask you the time we are in boarding school, when we are given meat, you find that the grade 12s, as we are in grade 8, they will end up giving you the one pieces which are appearing white, and they usually used to be bigger than ligaments. So that when you are chewing, they are always stretchy, that when you stretch, sometimes it might let go and come back and hit you in the face with all that soup. Or maybe you are pulling, it comes out and splashes soup on your friend. So that is the one which is stretchy. So the one which is stretchy, that is the ligament. But we also have tendons. Those are white. If you try to bite into it, it's very hard to bite into tendons. So that's why, but the ligaments are softer. They work like as if it's elastic. So those are the ones which join in bone to bone. So these are the ones which normally you are able to pull on your fingers. Those are held by tendons. But ligaments will join in the muscle to the bone, and those are very, very tough. So we also have the joint capsule. So this one will actually, the function of the joint capsule will be to enclose the joint membrane. So that will be the function of the parts of the synovial joint. Okay. So next we'll then move on to question number B, which says explain why a bone is regarded as a living tissue. Yes, guys. So why is a bone regarded as a living tissue? So the tissue will basically be secreted by the cells. So the cells will be secreting the fluid. That is where it's coming from. Okay, yes, commas are okay when you are dealing with the essays. That's fine. Alright, so a bone is regarded as a living tissue. So even if we look at bones and bones appear as if they are dead, they are actually alive. So what makes bones alive? One, they actually are made up of living cells. Then apart from that, bones also contain blood vessels. So there are blood vessels in there which are able to supply nutrients and carry waste from the bone. Another point is that bones are also alive because they are able to do what? To grow. So bones actually grow. It's very interesting that we actually grow a new skeleton every 10 years. We actually have a new skeleton altogether. So I don't know about you guys if you are skeleton number 1 or number 2 yet. So actually every 10 years you end up having a new skeleton, because the skeleton is always growing and replacing itself. Then apart from that, bones are also alive because they're able to produce red blood cells. So they contain red bone marrow, which produces red blood cells, yellow bone marrow, which will produce white blood cells. Right? So these are the reasons why bones are alive. Now, since bones are alive, just like any other living tissue, it can actually be infected by pathogens, and it can also have disorders. So tuberculosis can also infect the bones, meaning that you can have TB of the bones. If TB is not cured, it can actually infect the bones, and the bones end up swelling and have TB. Then you can also have bone marrow cancer, which of course referred to as leukemia, where you are now an individual produces a lot of immature white blood cells which are not able to defend the body against infection. So a person with leukemia ends up getting sick a lot. So this is how we present our essay. So we are saying bones are regarded as living tissues because they contain living cells, they grow, they contain blood vessels which provide nutrients to bone cells, and the bones contain red bone marrow which produces red blood cells, and the yellow bone marrow that produces white blood cells. Like other living tissues, bones can be infected by diseases. The common bone disorders are bone tuberculosis and bone marrow cancer. So this is why bones are regarded as living tissues. They have living cells, they grow, they contain blood, they produce red blood cells and the white blood cells, and apart from that, they can also become what? Infected. So mean that bones are alive. Okay, I hope that makes things clear. I hope you are clear on that one, guys. Otherwise, very good. So that is how bones are able to be regarded as living what? Organisms. Excellent work, guys. All right, moving on to question number C. Explain the action of antagonistic muscles of the eye when one moves from a dark room to a bright room. So what will happen to the eye or the pupil when one moves from a bright room to a dark room? Yes, guys. Very good. So when one moves into a bright room, the pupil actually becomes smaller. So when you're in a bright room, what basically happens is that the eye has got two antagonistic muscles in the iris. We have longitudinal muscles and circular muscles. So in a bright light, circular muscles will contract, while the radial muscles are going to what? Contract, which will then cause the pupil to do what? To constrict. So when a person moves from a dark room to a bright light, the excess light may damage the retina by bleaching it. So to avoid the retina breaching, that's why you find that, for example, if you are sleeping, someone turns on the light, you might start feeling pain in your eyes and you close your eyes because it's too bright. So to prevent the light damaging your eyes, what will happen is that the circular muscles will contract and the radial muscles will relax. So that will then cause the pupil to constrict and become smaller, preventing the bleaching of the retina by excess light. So when you come to write this essay, always make sure you start with the muscle which is contracting. So that's the first muscle you write. Then the muscle which relaxes, you write it at the end. So don't start the radial muscle relaxing and end with the circular muscles contracting. Always start with the muscle which contracts. Very good, guys. So that brings us to the end of our question number 9. We now move on to our last question, which is question number 10. Okay, so question 10 had three parts. A was saying describe factors that make soil fertile, 4 marks. B, explain the causes of loss of soil fertility. And C, explain methods of improving and retaining soil fertility. Okay. Alright, so we'll start with number 10A, describe factors that make soil fertile. So what makes soil fertile? Okay, so here you have to be careful. I remember the time I was teaching on this topic, I said there are 2 parts. There is the factors that make soil fertile and ways of improving soil fertility. So don't use the points for improving soil fertility. You bring them here for factors that make soil what? Fertile. Yes, guys. All right, so very good. Yes, so factors that make soil fertile. 1, we have soil air. So you need air in soil to actually make soil what? Fertile. Then apart from that, you also need the microorganisms in the soil. These are the ones which will also make the soil become what? Fertile. Apart from that, you also need moisture in the soil. That will actually ensure that the microorganisms are able to survive, and also water is a good nutrient for raw material for photosynthesis. Then we also need mineral salts in the soil. So these ones will be taken in as metal ions because they are needed by plants because they act as the activators for enzymes. Then we also need the dead organic matter. So we need the dead organic matter in the soil because it acts as the food nutrients for microorganisms. So these are the factors that make soil what? Fertile. Then also need the pH. Suitable pH also makes soil what? Fertile. Right? Very good. So these are the factors that make soil fertile. So factors that make soil fertile include soil air, soil water, microorganisms, mineral salts, dead organic matter, and the pH. So soil air is needed for respiration to release energy. Seeds need air for germination. Air helps the rocks to wither, and it prevents building up of poisonous gases. Then microorganisms such as fungi, bacteria, and algae play an important part in soil fertility and the nutrient recycling. So moisture is necessary for photosynthesis to take place in plants, and it dissolves mineral salts before they are absorbed by root hairs. Mineral salts are absorbed as the ions by the root hairs of plants. They are needed for plant growth. Then dead organic matter supplies food to the soil organisms such as earthworms, ants, and the rodents. Then pH affects the availability of mineral salts in the soil. Acidic soil contains very few mineral salts and it kills the microorganisms. So that's why having acidic soil is bad. Right? So that is the factors that make soil what? Fertile. Okay, so don't confuse them with the ways of improving soil fertility, like adding fertilizer, altogether weeding, crop rotation. So that is why we improve soil fertility. So next it says explain the causes of loss of soil fertility. So how does the soil become infertile? What causes the loss in the soil fertility? Yes, guys. So what will cause loss in soil fertility? One, we have deforestation altogether. The cutting down of indigenous trees will lead to loss in soil fertility. We also have poor farming methods can also lead to loss in soil fertility. We also have overgrazing altogether. Apart from overgrazing, we also have late burning, and apart from late burning, we also have harvesting can also lead to soil fertility. So the answer will be as follows. Soil fertility is caused by the following factors, namely deforestation, poor farming methods, overgrazing, late burning, and the harvesting. So deforestation leads to washing away of nutrients, decrease in soil organisms that make soil fertile. Poor farming methods such as adding too much inorganic fertilizers to the soil can change the structure of the soil, reduce pH, making the soil acidic, and increase soil erosion. Then late burning destroys seeds that fall to the ground, kill microorganisms in the soil because the heat is too much. That makes soil fertile by decomposing organic matter. Then overgrazing will cause leaching. So leaching is the draining of soil from the leaching is the draining of water from the nutrients, washing away with water in sandy soil. So leaching is simply when it rains, the water will dissolve the mineral salts, and as the water flows, it also carries away the mineral salt. So that's why overgrazing is bad. That's why when you have a farm, don't just put a lot of cows or goats on the same area. You need to have a certain number so that you don't have overgrazing taking place. Right? Then harvesting will remove the nutrients used by the plants to grow. So when the plants are growing the soil, they are sucking up the nutrients. Right? So once your vegetables grow nice and big, when you get your vegetables, you've also carried away the nutrients which the plant had sucked up. So when you're eating your vegetables, you're eating your nutrients. So that's why the soil becomes infertile. That's why after you harvest, you need to remand the soil again, add more manure, so that the next growing session has the nutrients again. Alright. So next we now move to number C. So this is the part now which I also say ways of improving soil fertility. Right? So and the retaining fertility. So normally people confuse the points for factors that make soil fertile and the ways of improving soil what? Fertility. So what should be done to keep the soil so that the soil keeps on being what? Fertile, right? Yes, guys? Very good, yes. We need crop rotation to improve soil fertility. Very good. So those are the points which we need. Any other points which we might need to make and improve soil fertility? All right, so what do we have? We have adding of lime now together. So we can add lime to improve soil fertility. This is when we are using organic fertilizers because the soil becomes acidic. So when you add lime, it neutralizes the acid. Weeding is also another way in which you can improve soil fertility because the weeds, once they are removed, they reduce competition with the plants because the weeds now don't take up the nutrients from the soil. Apart from that, we can also add fertilizer. It will also improve the soil fertility. We can also practice crop rotation, which you stated there correctly. So one season you plant maize, the other season you are planting legumes like groundnuts or soya beans or beans itself. So keep on rotating your crops. So that will improve soil fertility. Apart from that, we also have conservation farming. So instead of us, when we cut our maize, let's not move the maize cobs out of the field. Keep them in there in the field so that they decompose, adding nutrients again back into the soil. Right? So that is what we refer to as conservation farming, where you conserve everything. You don't throw anything away. Everything goes back into the soil again. Okay? So this is how we are going to write our essay. So soil fertility can be improved and retained by the following methods: adding lime, weeding, application of fertilizer, crop rotation, and the conservation farming. Then we start explaining our points now. Adding lime is added to acidic soil to raise the pH. Lime can also provide crabs to retain water and supply nutrients to plants. Weeding regularly ensures more nutrients, water, space, and oxygen for growing of harvest crops. Application of fertilizers retains nutrients that have been lost from the soil, essentially for growth, for plant growth. Sorry, essential for plant growth. Get it right. So crop rotation with the legumes to fix nitrogen in the soil with the nodules on their roots, which replace nutrients in the soil. Then we are saying conservation farming is important to sustain soil healthy and fertility and to provide enough food for growing populations. So this is how we are able to improve our soil fertility. So all these notes are there in the lesson which I gave last time when we were looking at soil as a topic. So this is how you answer your questions in biology. So we've done all the papers. So now once you are done answering your options, there's this box which you should never forget about. So when you're done answering, here it says number 2. There's this point I don't want you guys to forget. Enter the number of section B questions you've answered in the grid on the bottom right corner. So section B, if you answer your first question as question number 10, you write it down here. Where's my marker? You write it down here as 10. That was the first one you answered. So when you answer in section B, you can start with any question. You're not fixed to answer them in order. So whichever one you find you've understood the most is the one you start with. Right? Then if the next question you answered was 6, you write your 6 down here. Where's my mark? Oh, here. So you write it as 6. Then in your last question, if it was 7 or 9, you write it down here as 9. So always make sure you fill up this box. Majority of the time when candidates write biology, you normally don't fill this box. So it's very important that you fill in the question numbers you've answered. Okay, guys? Alright, so thank you so much for watching, guys. This is the end of our stream. Otherwise, I really enjoyed your company. If you are still here, well done, guys, for making it all the way up to the end. Thank you so much, guys. I really appreciated your company, and thank you. It's been a long stream and you've been participating fully. So thank you so much, guys. Really appreciate this. Hopefully we've understood 1 or 2 things which we've learned actually today. Otherwise, thank you so much once again. Otherwise, I've been done with the flu, but your encouraging words have actually helped me push through, that we've been able to do this question all the way up to the end. All right. So for now, I'll simply say good night. Thank you so much, guys. May God bless you too. and enjoy the rest of your evening. I know it's almost 2022 now, so have a blessed night. As for now, I'll simply say good night and God bless. See you in the next one. Thank you so much for